Hey, welcome to Reproduction. I just want to go over a few things briefly to help you focus on what to learn and what to really incorporate. The first thing I'm going to do is male reproduction, and then I'll go into female reproduction. I think this personally is a really good figure for you to study from. Not only does it show you the meiosis that's occurring, which you do need to know, but also the hormonal endocrine um, interactions, as well as how that maturation of the spermatogonia and then subsequently the spermatocytes and spermatozoa happen, and then where it's housed, so to speak, before ejaculation occurs. And so I would make sure you understand this diagram really well. I will tell you that you should know the differences between meiosis in the male and the female. They are different, even though we learned very typical in biology, beginning biology classes, you learn the very standard mitosis and meiosis. In actuality, female meiosis is quite different for the most part. In males, it's pretty much the classic case where you get from one sperm cell, and as you can see, you'll have a division of that stem cell. If I can get my green arrow, there it is, to come out. Oh, maybe. Okay. One then divides, and then as the daughter cells, you end up getting essentially from one stem cell 4 spermatotids or spermatozoa for the most part. It's a 1 to 4 thing. It's once it starts, it continues. So once this starts in the male, the initiation of puberty, it will continue until he dies. There is a peak period that occurs that's related a little bit to the hormones, and then it will taper off, but theoretically a man in his 90s is still producing sperm. So, there's some things we'll talk about in female that are a little bit different. You do need to know the differences, and you need to know the terms that are used, the primary spermatocyte, secondary spermatocyte, a spermatid, and a spermatozoa. You should know those terms. The other thing about meiosis here is that you have a couple of cells that you see here, a cystenticular cell, or what we sometimes have referred to as a Sertoli cell or a nurse cell, helps provide the nutrients and the growth factors necessary for the stem cells to divide and for the spermatogenesis to occur. In addition, you have in the testes, you have Leydig cells, and those Leydig cells release. So here's an example of one right here on the outside of the seminiferous tubule. Leydig cells respond to LH and release testosterone. You do need to understand the hormone interactions with and what's happening in this area. So FSH stimulates spermatogenesis. LH stimulates testosterone. Testosterone does give a negative feedback both to the pituitary and to the hypothalamus. And also inhibin is produced here in the seminiferous tubules to also give a negative feedback. And so you should understand the role of these reproductive hormones and what they're actually doing. As you can see, this is just simply a cross-section that occurs in the seminiferous tubules. These where spermatogenesis occurs. It goes from a basal layer, and as they develop and as they grow and mature, they go into the lumen. So you see this seems people sometimes think of it sometimes backwards, but it actually starts here at the basal layer and then moves forward or moves inward into the lumen. And then this process of producing spermatozoa takes about 10 weeks. So you have the large bulk of that time is in the seminiferous tubules, and then as they mature and grow, they go into the epididymis. And in the epididymis, it's 10 to 14 days before final maturation takes place. Prior to this point, the sperm are unable to be motile. I mean, they're inhibited from motility anyway, but they're not functionally able to be motile, so they're not really any good until they get to that point, if as far as any type of natural conception goes. And then from the epididymis, they are released through the ductus deferens and out through the male urethra, and there's a separate figure for that. And a separate physiology of what happens with erection ejaculation. I think what you should focus your time on, though, is understanding, once again, spermatogenesis, the role that LH and FSH have in the male, and of course, LH and FSH are stimulated by gonadotropin-releasing hormone, which you should have learned in the last module, which is further stimulated by well, gonadotropin-releasing hormone comes from the hypothalamus, stimulates LH and FSH, from the pituitary, anterior pituitary, and then comes to the seminiferous to the testes. The testes is actually the male sex organ. A lot of individuals get that question wrong. I don't know if it's on this particular exam. It will probably be on one of your quizzes, though. Often, incorrectly, it is thought that the penis is the male sex organ, but actually that's the conduit to where the sperm can be ejaculated. The organ, the one that actually produces the sperm, is the testes. And so please make sure you have that distinction. Once again, the hormones have the effect on the LH cells will release testosterone, which aids in the continuation of spermatogenesis, but FSH is really necessary in order to get that process started and going. And so both of them have a function in male as well as female. And once again, that feedback. The other thing that's probably a good thing to emphasize is to talk and at least understand what's involved in the seminal fluid. So this isn't a picture of seminal fluid, but you need to understand the glands that help contribute to semen, which comprises of sperm themselves and of various contributions from the 3 accessory glands. We have the seminal vesicles, which contribute about 60%, the prostate gland, which contributes about 30%, and the bulbourethral gland, which contributes about 5% of the overall volume of semen. Now, normal semen volume is considered anything 1.5 mils and above. Typically, that's what you see is 1.5 mils to 2 mils, and if you have a small syringe, that's, you're looking at at the most half a teaspoon. A teaspoon's 5 mils, and that's a fairly large quantity of seminal fluid. Actually, we don't see that quite often. I would say it probably averages about 2.5 to 3 mils, and so you're looking at essentially half a teaspoon is considered normal volume in a given semen sample. So you should understand those contributions, and also a little bit about the sperm themselves. So in the male, it's a quantity versus quality type of issue. So you may have hundreds of thousands or millions of sperm coming out per ejaculate, and that's all fine and good, but they're all not necessarily perfectly shaped or moving well or that type of thing. And so count is really our least likely predictor of success when it comes to an analysis of sperm and of the seminal fluid and what's happening there. There are various things that we measure. We measure movement. We measure morphology as far as tail shape and head shape. Those are actually checked differently in some cases, depending on which clinic you go to. But that's checked. The viability, how the membrane function structure is, that is called. We have viability and membrane function tests. I guess they're 2 separate tests, but you even go so far as to find out if the acrosome reaction is working properly and everything in sperm. And so even though there may be, once again, millions of sperm, not all of those are going to be really qualified in order to try to penetrate the ovum, which is their, sole purpose in life. And so they all are haploid cells, as you all should know, and go through that fertilization process. We talk about that after we talk about female physiology. But so you should know general characteristics of seminal fluid, the contribution, and what that seminal, for example, seminal vesicle, 60% of that fluid. All right, well, what's so important about that fluid? Well, one of the important components, and probably the one that we care about the most, is fructose. And fructose is there to help with the mitochondria that's wrapped around the tail. And mid-piece, you can actually see it's kind of shown in, this is a rudimentary drawing here, but kind of shown in brown here. Mitochondria is wrapped and coiled around here in this mid-piece area, and that's what helps break down the fructose to give the energy to the flagella, which then becomes the tail and causes the propulsion of the movement. And this movement changes as it gets closer to the egg. It goes from a fluid, what we call progressive motility, where it's a straight line, it's moving fairly straight line fairly rapidly, to more of a jerking motion as it's undergoing acrosome reaction and capacitation. And so There's a lot of different characteristics that are important in order to really understand how fertile a man is and whether or not that semen sample seems to be within normal range of what one would expect for someone who could fertilize and impregnate um, his partner. So um, seminal vesicles contain a lot of fructose, just as an example of one of the things. Prostate gland contains antibiotic-like factors and also helps with buffering. The vagina is in a very acidic environment, and so in order to protect the sperm, we have to have some buffers there. And then the bulbourethral gland secretes somewhat lubricates actually the reproductive tract, so the sperm can exit appropriately and without problems. And so each of those has a contribution which you should understand. Okay, enough of that for now, because like I said, I did want it to be kind of brief, so you're not listening to a lot. I could talk for a couple hours actually about sperm, since that is my field of study and for my career as a male fertility clinic at the University of Utah. And so I'm not going to do that anymore, but I do want to spend, if you have other questions, you're welcome to email me, or if you have questions on your quiz you don't understand, please email me. But I do want to talk about female reproduction. And I think female reproduction is actually broken down into 3 different cycles. The uterine cycle, the ovarian cycle, and the hormone cycle. And so let's take a look first at the ovarian cycle. This is the ovary. The ovary is the female equivalent of the testes, of the male testes. So the ovary is the female sex organ. This is where oogenesis, or the production of oocytes, a mature follicle that can be potentially fertilized, happens. As I mentioned, the difference between meiosis in men and meiosis in women as far as sperm and eggs go, the female reproductive cycle is really unique in the sense that women in utero, actually, so as a fetus, meiosis begins in the female and then it stops, and it's arrested, so to speak, at a very early stage, and then it starts up again at puberty, and you go into the first finishing of the first phase of meiosis, and then after that, that occurs actually at ovulation, where the first cycle of meiosis is complete. And the second cycle of meiosis doesn't actually complete unless the egg is fertilized. And if it's not fertilized, it really never completes meiosis as we learned it in biology. And so make sure you spend time understanding that. I did not see offhand a diagram that talked about that, but you should be able to find one. Let me just double check. It might be embedded in the thing. Let me just look really fast. Sorry. I should probably just pause. I don't see it. So I'm just going to go on. So the ovarian cycle this is what we talk about when we talk about a woman's cycle and how many days it is. This stuff is happening both here and in the uterus and the hormones simultaneously. And so you kind of have to get all 3 pictures into one, and I'll show you another diagram a little bit later. But the first couple of days, due to the presence of FSH from the pituitary gland, due to the stimulation of the gonadotropin-releasing hormone, and due to the lack of inhibin and lack of inhibition at that site, we see the first couple of days a rise in FSH, or follicle-stimulating hormone, which does exactly what it's referred to. It starts stimulating about a dozen follicles, and they all start growing. This picture only shows one, but there's several here that start growing and developing, and as they grow and develop, and you see they start getting layers of cells around them and forming this follicle with some fluid in it. And at some point in time, a primary follicle is chosen. I don't know if anyone's noticed or talked about this, but if you get a group of women together, one woman eventually will become, through pheromones or through hormone release, the dominant female, and the other females will actually end up matching that cycle of the dominant female. It happens in the animal kingdom all over the place, but it happens with humans too, actually. And so if you have a dorm with a bunch of women, then you'll find after several months that a lot of them will start cycling at the same time, and that's because there's a dominant female there that's like I said, releasing some types of pheromones or something that the others somehow, it's still kind of a mystery, but they seem to sense to follow and sink in. The same type of thing's happening here. So you have a group of these, 10 to 12 follicles are all kind of developing. One of them's just a little bigger. One of them's getting a little bit more of that hormone, and then because of that, they're making more receptors for LH, and they're making some estrogen, and that estrogen then helps kind of feed and mature and grow this follicle. And so one becomes a lead, or what we call a primary follicle, and all the rest ultimately are contributing hormones to help the primary follicle. In the male, remember I told you it's kind of a quantity versus quality. In the female, if you want to look at it in a different aspect, it's like an all for one, like a Three Musketeer type of thing. All for one, one for all. So the lead follicle gets all the nutrients, gets more and more of that thing, and the rest kind of end up fading away or going through atresia is the official term for it. Here's an example of the follicle atresia that may be occurring with other follicles. Okay. Anyway, so you go from a primary follicle to a secondary, and then as you grow mature, you get this liquid-filled space. It's called an antrum, and it's filled with what we call follicular fluid. Really creative there, but it actually has nutrients and other factors we think that are really important for proper development, not just of the follicle itself, but after fertilization. So if you're doing advanced reproductive techniques, we mimic the fluid properties that are here in this area. And of course, some of that fluid is exuded normally in a normal fertilization process, and seems to help this mature follicle. As this process is going, and as estrogen is increasing and continuing that development, and we'll look at a hormone chart in a minute, you get to the point where the conditions are such that you're going to see just this surge of very high quantity of LH from the pituitary be released. And that does two things. It helps the final maturation of this oocyte, and it helps with ovulation, which is the release of the follicle from the ovary, and which goes into the fallopian tube and hopefully down the track, in this case, has fertilization occur, because that's its whole purpose of this process is to have fertilization and to create an embryo and then subsequently a fetus and a baby. The leftover follicle cells that remain in the ovary develop into the corpus luteum, and this is a hormone-producing body. It will continue to do so until the placenta is formed if it receives a signal. That signal is HCG, that's a pregnancy hormone, that the little embryo is sending up. And it will, like I said, once again, it will function until the placenta can take over. If there isn't fertilization, and if there isn't an embryo to send a chemical hormone signal of HCG, then after about 10 to 12 days, this stops producing progesterone and estrogen and starts essentially to disintegrate, die off. And these are the names of it, the corpus luteum and then the corpus albicans. This is known as kind of a yellowish structure with all the cells and the secretion of those hormones, and the corpus albicans is more of a little white structure. Anyway, if estrogen and progesterone aren't being produced by the ovary and they're not being produced by a placenta, then there's no reason to have the endometrial lining. So a woman will have her period and the process begins over again. So this is the ovarian viewpoint. Let's look at the hormone viewpoint. And the uterus viewpoint is in with this diagram. The one that they selected for the female cycle has this both. So let's look at this. And this is probably the most important aspect of female reproduction to really focus on, is kind of getting this all together. So let me show you here. This is the ovarian cycle, once again trying to show you that there are phases, what happens here. These follicles then develop, and it says it develops for 185 days. That's because there's always a bunch of little ones working towards it. In a cycle, when we have a primary follicle, or in the sense of having a primary follicle, meaning one that has been selected and goes into the stages of growth that we just saw in the ovary, that happens approximately once every 28 days. Okay. All right, and this is just kind of once again showing the size of what happens, and ovulation occurs in corpus luteum, and see how it turns white if you don't have hormones. Okay, this is the uterine cycle here. This first 1 through 5 days approximately is considered a menses, or a period where there's a sloughing off, where the blood supply and the tissue is released from the uterus in the presence of increasing levels of estrogen. Then we start seeing it build up again, and this proliferation phase, you do a lot of mitosis division, a lot of building up. And then in the secretory phase, a lot of blood supply comes through here. This is usually due to progesterone. We get it very rich with a blood supply and availability, ready to accept an implanted embryo or an embryo to implant. And if it does, then these levels of progesterone and estrogen remain high until the end of pregnancy. If not, then they start to lower as the corpus luteum no longer functions. And then when they get to a certain ratio, then once again you see a sloughing off. And so there's 3 phases to the uterine cycle. The hormones, it's kind of hard to see a little bit here because they've been superimposed on this diagram. But the ones from the pituitary are up here, LH and FSH. High FSH for a couple days, and then it kind of stays lower. LH stays pretty low, does this surge or this large peak, and then as it comes down, ovulation occurs. Because of this rise and because of the influence of stimulation of LH, we see a slight increase in FSH as well. They both drop and they stay low. Once again, the FSH will start to increase as the progesterone and estrogen drop and change the ratio. Notice progesterone's higher than estrogen here. Estrogen's in red, progesterone's in black. And as this gets to this point, we start seeing an increase of FSH due to a lack of inhibin. Here's another showing of inhibin dropping as well. Inhibin does the same thing in the male as it does in the female. It inhibits the release of the gonadotropin-releasing hormone and has that effect on the hypothalamus as well as the pituitary, actually. Notice progesterone is only made post-ovulation, very low. This is really basal line. And then due to the corpus luteum is when we see progesterone. Estrogen, on the other hand, is being produced and developed from these follicles and all these little supporting follicles. And so you see a rise in estrogen as this follicle grows and develops, and as the other ones contribute to that growth of that primary follicle. And so you see a rise in estrogen. It does drop because that follicle has been released, but then it increases a little bit due to the corpus luteum. And once again, if pregnant, you'd see these, instead of having humps here or having bumps, they would just kind of stay pretty much straight across until it was time for the baby to be born. Okay, they brought up basal body temperature. That's really not a good predictor of a potential ability of pregnancy. It used to be the way that women would test to see if they were ready to try to achieve pregnancy, because you'd see a slight drop and then you'd see a sharp rise. Notice that the thing's only 0.6 degrees, and a lot of that can be affected by other things, and so it's not a great predictor. Certainly not like testing one's urine for LH, which is what's commonly done now for women that are trying to sync their cycles a little bit better and trying to achieve pregnancy. Ovulation predictor kits are really looking for this rise, knowing that this surge is for 36 hours. And so if you catch it right here or catch it on the downswing, it's safe to say within 24 hours, depending on when you're testing, is when your egg is released. And why is that important? Well, the secondary follicle, once again, undergoes that first set of meiosis or first stage of meiosis and ovulation, but is really only viable for about 24 hours in order to achieve a pregnancy. It does live a little bit longer than that, but it doesn't have the time to develop, divide, implant, and release HCG in order to keep the corpus luteum going. And so it really only has about a 24-hour window. Male sperm, on the other hand, can live anywhere from 3 to 5 days in the female environment, and so that's why you see a larger range of potential pregnancy days when you're either looking to be pregnant or trying to avoid pregnancy naturally. There's more days because the sperm do live in the environment, like sitting anywhere from 3 to 5 days. We find out that they have a tendency to hang out in the curves by the fallopian tubes and in the uterus, and they'll they'll hang out there for a couple days and then move forward again. A lot of their drive potentially can be used from cytokine chemicals. It's thought that the egg sends out signals, so to speak, so they know kind of where they go through both fallopian tubes, even though one's only contains the egg. But there's also an idea that maybe there's some chemical releasing there that will be attracted by the sperm, that they'll move towards there. And maybe that's why they also hang out for a few days, is because they're not sensing those chemicals. And so there's still a lot of work and research to be done both in male and female reproductive physiology. Most of it's with protein, genome, proteins and stuff like that. But anyway, don't want to get too much into the woods there. All right, let's see if there's anything else I want to share. Let's see. I don't even know what that is. I don't want to pull it up if I don't know what it is. Let's look at this one really quickly. You do need to understand the anatomy to understand the physiologies, but I wouldn't focus super hard on the anatomy. I'd focus more on meiosis in male and female, the ovarian cycle, and how it correlates with the uterine cycle and the hormone cycle. That's really where you're going to see the large bulk of your focus on your exam questions for both male and female. In the male, also sperm characteristics and stuff like that. Oh, this is an old picture. Just kind of showing we typically want a fertilization to occur in the fallopian tube. It takes, once again, the egg's only viable for about 24 hours, and it takes another 24 hours or so to have that first division. And then you want to have it at about the 5th day post-fertilization. By that point, implanting into the uterus. By day 7, the embryo will be releasing HCG, which then causes that corpus luteum to continue producing. And so a lot of things can happen in between that stage. It could be the sperm just don't penetrate the egg, or the egg's wall is like the zona pellucida, which is the wall around the oocyte or egg, is like a big 30-foot wall of cement. So sometimes male sperm can't get through. Other times maybe it's even harder. It's supposed to be a little bit more pliable. In younger women we see that actually. In other women and older women it's a little bit harder. Of course there's genetic potential issues. So there's a lot of things that can interfere with this process of fertilization and implantation in order to achieve pregnancy. And we could spend a lot of time talking about that, but we won't. So that's just really basic review and male and female physiology. Mainly just wanted to give you some of the basics to focus on. I will try to give you a little bit, I'll do a really short screencast with just focus on for your exam. Instead of, we don't really have time for an exam review because you finish Thursday night and Friday the exam opens. And so I know most of you won't take it on Friday, but for those of you that are, and especially with General Conference this weekend, maybe wanting to focus on other things this weekend, your test does close on Monday. But I'll do a short screencast to help you focus on your last exam for endocrinology and reproduction. So look for that in the announcements. Thanks for spending the time, and have a good night.